A developmental network would be the worst television. Hey there, today we're going to talk about the genetic developmental network that basically explains how genes interact with one another and how they create a certain phenotypic output. In our hypothetical example, we'll show how formation of spikes in certain cells is created using this developmental network in the thorny dragon lizard. By the way, just want to clarify, I love animals, but only a mother could love that. Here we can see the developmental network for thorny dragon spike. We can see the 5 genes S, P, I, K, and E that are all involved in the formation of spike. We know that these genes are all associated and have relationships with one another. We can read these relationships by looking at the symbols in between each gene. The red arrow means that one gene activates the other, and the T-bar shows that one gene inhibits the other. Now we'll look at different mutations of the developmental network and see if they produce spikes. If we look at the wild type, which means there's no mutation, we see that S activates the gene P, and P inhibits I. So since I is inhibited, it cannot inhibit K, which means that K activates E, which means that activates the production of spikes, which means in the wild type mutation there will be spikes. Now we look at the developmental network with a P mutation, which means that the P gene will not express its usual behavior. So its usual behavior would be inhibiting the I gene, which it won't do. If it doesn't inhibit the I gene, that means I can inhibit K, and K cannot activate E, meaning that there will be no production of spikes. Now we look at the I gene mutation, which means that it won't express its original behavior of inhibiting K. So we look at the developmental network and see that S activates P, but P doesn't have anything to inhibit because I is gone. So that means K is allowed to activate E, which means it activates the spike production in those lizards, causing spike formation. Now we look at the mutation of the I and E gene. Just like the last example, I cannot express the inhibition of K, leading K to activate E. However, E cannot express the activation of spikes because it's mutated. So that results in no spike formation. Here we have is the S and K mutation. We see that S is mutated where it can't activate the P gene. But what really matters is the gene mutation downstream because it has more of an effect on the phenotypic outcome. We see that K is mutated where it can't activate the E gene. If E is not activated, that means that it can't activate the production of spikes, meaning there's no spikes. One of the biggest questions is, how come not all cells in the skin have spikes even though they have the same genome sequences? A possible answer could be gene regulation, more specifically DNA methylation, which causes cell differentiation. DNA methylation regulates gene expression by recruiting proteins involved in gene repression or inhibiting the transcription factors of DNA. During development, the pattern of DNA methylation in the genome changes by preventing transcription and repressing gene expression, which results in types of cells, cell differentiation, which is why some cells might have spikes and some cells may not have spikes.